All right, good afternoon, everybody, and welcome to today's webinar sponsored by NAVBO's Education Committee. I'm Dr. Joshua Scallon from the University of South Florida, and I'll be moderating today's session. I'm pleased to welcome Dr. Josh Wythe from the University of Virginia and Dr. Zhanna Nepiyushchikh from Georgia Institute of Technology, who will present their talks from the 22nd International Vascular Biology meeting. Before we begin, I'd like to remind you that all NAVBO activities are easily found in the calendar of events on our website. Please be sure to register for some of these upcoming events. Throughout each presentation, please type your questions in the question box. You don't have to wait until the end of the talk. You could just type it in as soon as you have a question. That way you don't forget. And these questions will be answered at the end of each talk. This webinar is being recorded and archived on the NAVBO website for future use. First, I'd like to introduce Dr. Joshua Wythe, who will present Targeting the Cerebrovasculature to Combat Glioblastoma Multiforme. Dr. Wythe is a tenured associate professor at the University of Virginia School of Medicine, and a member of the Robert M. Byrne Cardiovascular Research Center and UVA Comprehensive Cancer Center. Dr. Wythe's laboratory studies human diseases by generating novel animal models using both zebrafish and mice, with a specific focus on the cerebrovasculature as well as developmental vasculogenesis, angiogenesis, lymphangiogenesis. After completing his graduate training with Dean Lee, who's the current president of Merck Research Laboratories at the University of Utah, Josh performed his postdoctoral work at UCSF in Dr. Benoit Bruneau's lab, where he studied heart field lineage allocation and the transcriptional basis of arteriovenous specification. There he was promoted to non-tenure track assistant professor, and after receiving an AHA Career Development Award, he joined the Cardiovascular Research Institute at Baylor College of Medicine, where he was a tenured associate professor. His lab has made seminal contributions to the field's understanding of somatic brain arteriovenous malformations and continues to develop novel technologies and imaging methods for studying the cerebrovasculature. Their work has been funded by the Department of Defense, NIH, Canadian Institutes of Health Research, Cancer Research, and Prevention Institute of Texas, and through various private mechanisms. So with that, I'd like to welcome Dr. Joshua Wythe and hand it over to him.
Thanks, Josh. I appreciate that. Before I begin, I'd like to thank the NAVBO Education Committee for giving me a chance to share with you some of the work that my lab at the University of Virginia School of Medicine has been pursuing on targeting the cerebrovasculature to combat GBM. So glioblastoma multiforme, or GBM, is an aggressive tumor of the central nervous system that arises from glial cells. Though relatively rare, it is the most common primary brain tumor among adults, with about 20,000 cases in the US annually. Now, GBM, unfortunately, has a 5-year survival rate hovering right around 6% and a median survival time of approximately 8 months. Now, critically, survival rates for GBM have not changed in the past 30 years, and this really highlights the need for new approaches that are basically really desperately needed to combat this devastating disease. Now, as you can see in this image, where the lesion or tumor is marked with a green arrow and vessels are highlighted by an angiogram in the image below, there's extensive vascularization around the tumors. You compare the side with the tumor to the contralateral, the ipsilateral side here. And as we zoom in, you can see that there's basically intense signal leaking around this, which is evident of a leaky vasculature. And this is more obvious when we look at this coronal slab section here, where you can see that there's extensive hemorrhage here in the right side of the brain. Now, what's interesting is that GBM tumors that are wild type for isocitrate dehydrogenase 1 here are more proliferative and aggressive and have worse clinical outcomes, as you can see here where we chart survival, compared to IDH1 mutant tumors. Now, to get an understanding cluster normal in the normal resected areas featured an intact blood-brain barrier signature. And this is also present in the IDH1 mutant tumors as well. Again, these are the ones with the good or better outcome compared between the two. However, this was lost, this BBB-like signature was downregulated or lost in those more aggressive IDH1 wild-type tumors, as you can see here. Now, given the importance of angiogenesis and blood-brain barrier disruption in glioblastoma, we are interested in two key questions. The first of which is, can we manipulate the blood-brain barrier to improve outcomes in GBM? And the second one is, can tumor angiogenesis be disrupted to inhibit GBM progression? Now, to address these questions, we're using a immunocompetent native mouse model of glioblastoma. And what we do in this model is we take pregnant females around E14 to 16.5, and we'll expose the uterine horns during surgery. And we'll inject a DNA cocktail that targets tumor suppressors and also indelibly labels tumor cells with bioluminescent or fluorescent markers. So we inject that into the lateral ventricle of the brain around, again, E14 to 16.5. Then we'll electroporate the DNA into the cells of the brain. We place the embryos back into the female, and the uterine horn, they're still intact in there, and allow them to come to gestation. And then shortly after birth, we'll segregate these animals away after weaning, and later you will see a clear brain tumor or fluorescently tracked group of cells that are tumor or have these tumor suppressors knocked out. So the cocktail of genes that we target in this native model are three that are typically mutated by missense or loss of function or deletion mutations in humans: P10, P53, and NF1. And the rate of deletion is shown here. So these are three of the most frequently deleted genes in human glioblastoma. So the method that we use to knock these out in mice is a triple CRISPR vector, where we have Cas9 driven, and then in the same backbone, we have three guides, again, targeting P53, NF1, and P10. Now, to follow these cells over time, we use an early glial derivative promoter, one that will basically be expressing those cells, GLAST, in this driving piggyback transposase. Those of you who are more familiar with zebrafish, this is very similar to the TOL2 based system or transposase system. And then you provide a cargo plasmid that has ITR piggyback flanking sequences. So this cargo plasmid will actually be integrated into the genome, and you can label the cells with GFP, RFP, luciferase, whatever have you. So as you look at a mouse brain at P70, this is the olfactory bulbs to the cerebellum. You can see extensive tumor genesis here. If we take a section through there, there's by H&E histology, you can see cellular dysplasia and extensive proliferation. And in our triple Cas9 model, you can see that about most of mice, 50% or more, dead by postnatal day 100 after birth. So one thing that we see in this native mouse model, if we start the video here, where the vessels are labeled in gray and the tumor is labeled in green by the piggyback GFP, you can appreciate that the tumor itself is incredibly well vascularized as we zoom through the tumor using one of our more recent clearing techniques, Easy Clear, that we published in eLife not too long ago. And you can just appreciate, one, how diffuse the tumor is through the brain, also, again, how well invaginated the tumor is with the vasculature. At the same point, when we look between postnatal day 65 to postnatal day 80, so the tumor's labeled in green here on both sides, you can see the growth. When we do perfusion with Evans blue, which normally should not leak out of the vasculature into the underlying brain parenchyma, by postnatal day 80, you have extensive leakiness of the vasculature as this dye is extravasating into the underlying brain tissue. And this is quantified here between control and tumorigenic. So it's just GFP, IUE versus the triple Cas9 cocktail. And you can see how robustly this leak is happening. So it's indicative, again, of blood-brain barrier breakdown and in this model. So given the fact that we've got a model that has really strong face value or validity compared to what we see in the humans in terms of vascularization and a leaky BBB, these two questions are the ones that we're most interested in. again, that tumor angiogenesis could maybe be disrupted to inhibit GBM progression. And the second one being, can we manipulate the blood-brain barrier to promote improve outcomes in GBM? And for the sake of time today, I'm only going to talk about our efforts to combat angiogenesis in this setting. But before that, we need to have a better understanding so we're all on the same page about endothelial dynamics in GBM and what was known before we started this work. So interestingly, there's kind of two main mechanisms that could happen to control or basically stimulate angiogenesis in GBM. The first is probably what most people would think of, is where the tumor can secrete pro-angiogenic factors such as VEGF, and this results in local vessel growth from wild-type healthy vessels nearby. So we call these vessels that are recruited TAVs, or tumor-associated vessels, because they're associated with the tumor. At the same point, something that's really interesting about GBM, that's contentious when we started, is there's a model that glioma stem cells themselves can differentiate in endothelium and parasites potentially to give rise to their own vasculature. So we call these tumor-derived endothelial cells because, again, the endothelium that's derived from the tumor vasculature rather than vessels that are recruited nearby. So using our 3D imaging modalities and tissue clearing, we were able to see, again, in our model where all tumor cells are labeled with this piggyback GFP, we label the vessels with lectin here, that several vessels actually had these GFP positive cells, as you can see here. So this did show in our native mouse model of glioma that TDEX do in fact seem to contribute to the vasculature, at least the tumor stem cells can they themselves become part of the vasculature around the tumor. So with this in mind, we wanted to understand, do TAVs, those recruited vessels, and TDEX, those tumor-derived vessels, have a common molecular signature? Because if they do, then maybe that common pathway could be targeted to block tumor-associated angiogenesis and angiogenesis of those tumor-derived endothelial cells. And that's the key question we were kind of trying to tackle first. So to attack this, we took our, again, native IUE model of glioblastoma, immunocompetent mouse, and we excised the tumor. We did fluorescent activated cell sorting for TAVs, which are CD31 or PCAB positive, GFP negative, because they're not tumor derived, they're recruited, And then we also did FACS for those TDEX or tumor derived endothelial cells that are CD31 positive and GFP positive, and we performed bulk RNA sequencing. As you can see here in this heat map, where yellow represents upregulated genes and blue represents downregulated genes, when we look at several bulk tumor samples from postnatal day 60 and 80, they had a signature differentially expressed genes that identified or mapped to GO categories, such as multiple neuronal different processes, neurogenesis, immune system development, and neuroplastic cell signatures that we'd anticipate. Then when we look at the endothelium, those TAVs and TDEX, we see that they also have a distinct signature. As you can see that these enriched genes here are not enriched in the bulk tumor. So specific to the TAVs and the TDEX for pathways like Notch and Hippo signaling, as well as angiogenic processes like we'd somewhat expect, angiogenesis and blood vessel development. So it says that TAVs and TDEX do have this kind of angiogenic-like signature. Now, as we dug into the genes that were upregulated more, we became really interested because several of them we had identified before in another paper where we had treated endothelial cells with VEGF and profiled RNA transcription 1 hour later. We performed gene set enrichment data analysis of that HUVEC VEGF stimulated gene set compared to the TAV and TDEX gene set. We saw really nice enrichment or agreement between those. At the same point, when we used an existing CNS mouse angiogenic data set that's in the early developing brain, it's very angiogenic, we saw similar enrichment or agreement there. Now, this was really compelling to us because it suggested that TAV and TDEX really have this pro-angiogenic VEGF-like signature. So then we turned from our mouse and cell culture model to see what we could conclude from human data. So the Cancer Genome Atlas, or TCGA, had about over 500 bulk transcriptomes from low-grade glioma patients and malignant glioma patients. And when we looked at the profile of this genes that were in the signature in our mouse model, we saw that they were highly expressed in our GBM patients, but not in the low-grade glioma patients. As you see, there's more blue here and more yellow here. So suggested that the signature, again, is more associated with that malignant brain tumor rather than low-grade glioma. So this is interesting to us for a variety of reasons, because previously in the cell culture model, we, with Jason Fish's lab, had shown that VEGF through the MAP kinase cascade leads to phosphorylation of the ETS transcription factor ERG and subsequent recruitment of the histone acetylase P300. And then Bill Poo extended this work showing that ETS1 and not just ERG in a VEGF-dependent fashion also then recruits an epigenetic reader BRD4 to allow pause release of RNA pull2. So this is interesting to us, and we performed more ChIP-seq to look at these various chromatin remodelers and transcription factors in the absence and presence of VEGF. So we did immunoprecipitation for P300, HDK27, this epigenetic reader BRD4, and the ETS factor ERG. And what we saw when we mapped the chromatin occupancy of these various factors compared to ERG is really nice enrichment or colocalization of P300 and BRD4 on chromatin where this ETS factor already was. Excuse me. So the first step we want to look at then is see if multiple VEGF processes require BRD family function in vivo, because again, this has all been in vitro work. So we use a Miles assay where you can inject a vehicle IP into an animal, then about half hour later you inject Evans blue IV as a tracer, and then on back that's been narrowed and nuded, you can do increasing concentrations of VEGF, which should induce vascular permeability or leak, and then you can assay Evans blue leak outside of basically the injection site from the vessels, as you see here. And then you can compare this vehicle treatment to your drug of interest. In our case, we wanted to try a BET family inhibitor, which would target this BRD4 VEGF ERG pathway. And then you do the same analysis. And at the end, then you can compare your VEGF treated to the drug pre-treated with VEGF, and you hope that you get a lot of leak of Evans blue outside and less leak in your drug treatment, for instance. And then you can do a plate reader on this. So what we saw in our mice is if we inject increasing of concentrations of VEGF after perfusing them with Evans blue in the tail vein, you get this nice increase, again, in extravasation. However, if we pre-treat them with a BET inhibitor, that attenuates that vascular leak of the Evans blue into the skin. The results are quantified here. CPI 2 or 3 is the BET inhibitor that we use for these studies. You can see this various doses of BET inhibitor here. We're again able to diminish that vascular leak of Evans blue. At the same point, we are also interested in, okay, that's permeability and homeostasis. What about developmental angiogenesis? So we took a genetic approach where we took BRD4 flox mice that we engineered in the lab, coupled with CH5 PAC-3 ER from Ralph Adams' lab, and injected pups at postnatal day 1 and 3, and then harvested retinas at P6 to look at angiogenesis in postnatal retina. So you can take the eye and excise the retina, which is a 3D optic cup. We make 4 radial incisions, and you can actually track angiogenesis in a very stereotypical way across developmental time. So using this model, what we saw in a BRD4 inducible endothelial cell knockouts was there's an increase in the avascular space, so basically radial expansion of the vasculature, and that leaflet at postnatal day 6 was severely blocked, as you can see here compared to the controls. And also, if we zoom in the vascular front, we see a real absence of filipodia and lamellipodia, which are here, you see indicative of angiogenic sprouting. This is just quantification of radial expansion, as you can see here, again, at postnatal day 6. So with this evidence from cell culture and all the data that we've seen in our native model of glioblastoma, that there's a pro-angiogenic signature and demonstration that BRD4 seems to be required for VEGF-dependent processes of vascular leak and developmental angiogenesis, we wondered again if this could be targeted to block angiogenesis in this disease, pathologic angiogenesis, and if it could have any positive impact on tumor growth and survival over time. So we took our IUE model at 16.5. We let the pups come to birth that have been mutated or knocked down for these 3 tumor suppressors. We screened them by bioluminescent imaging at P35 to P55, and then we actually treated with a frontline chemotherapeutic as well as an inhibitor of that family members in this window. We imaged them over time to track tumor growth, as you can see here, from 35 to 55, 75, 95, where the tumors are bioluminescent here. And then we let them also go to survival after we end that treatment. and while it's still early days and underpowered a little bit, what we can appreciate is about 50% of survival treated or untreated in vehicle. You get about half of them are dying somewhere between P60 and P80, right about here. And there you see the time to death. And our TMZ, the frontline chemotherapeutic, has a substantial, significant increase in survival time at P50, as well as to death. And OTX-015, which is our blood-brain barrier permeable BET inhibitor, as well as a combination of TMZ and OTX, seem to confer some increased survival relative to untreated mice, although it does not appear that the two drugs together, as you see here in green, confer increased survival over either drug alone. And this might be due to the toxicity of combining these two really honestly toxic compounds. So I hope today that I've convinced you that tumor vessels are extensively remodeled, as you saw the blood-brain barrier leak and the angiogenic signature that we were able to identify, and that TDEX rarely but consistently contributed to tumor vasculature, as we showed you with our light sheet 3D imaging data. And furthermore, a native murine model of glioblastoma with our IUE and patient-derived xenograft that I did not have time to show you today, and TCGA and our early single-cell data all show a pro-angiogenic anti-BBB signature seems to correlate with high-grade glioma, but not low-grade glioma. In the future directions where we're headed right now, we're trying to verify if disrupting BRD4 activity affects the angiogenesis and tumor progression in vivo. So with that, I need to thank the work, or the people who actually did the work. Right now, a very talented technician in the lab, William Gillespie, has been leading the drug studies here, and a previous graduate student in the lab, Manuel Cantu Gutierrez, led the 3D imaging and initial transcriptional characterization of the native model of glioma, and he was helped by William Turner, a former postdoc lab, Alexander Herman, and two other technicians, Sebastian Calderon and Nicholas Hudson. And with that, I'll thank our funding sources from the past, like CPRIT when we were in Texas, and now at the University of Virginia, and I'd be happy to take any questions. Thank you.
All right. Thank you, Josh, for the excellent talk. So at this time, if you have any questions, just feel free to type those in the question box. I have a few questions myself while we're waiting. I think your video may have frozen a little bit at the very beginning of your slide, so I might have a few questions that maybe you might have covered, but it was really brief. Okay. But I mean, just also maybe just for myself. So why are you doing intrauterine injection instead of postnatal injection into the mouse brain? Is that because there's bone covering? It's hard to inject through bone, or do you think the intrauterine injection is mimicking the timing of the genetic mutations in the patients that develop glioblastoma?
I think that's a great question. So you can do injection stereotactically into the brain postnatally. In our hands, it's not quite tolerated as well, and then it would also probably be location dependent. So we think it's easier to hit some of the progenitor cells earlier with this, and that it does have the length of time to accumulate those mutations is appropriate, because it's pretty close to birth, right? Because even though we electroporate at like 16 or so, you know, they're born 3 to 4 days later because the surgery affects their gestation time. It's not like the P21, P22 time to birth. But we think it's a reasonable model for this, but you could do that. But I can't comment on how well the electroporation would work. Certainly with lentivirus, you could do it, but then you've got the risk of genome integration later, where these plasmids stay predominantly episomal. But our piggyback ones certainly do integrate.
Yeah, okay, cool. We have a question now in the box. So David Gordon's asking, do the treatments which prolong life in your mouse model also result in a lower proliferation rate of the tumor cells?
That's a great question. So our preliminary data is, again, you know, these are all like cohorts of 12 to 15 animals per treatment group, is something I didn't want to get into just for sake of time, is that we see there's variability in terms of the size of the tumors. So we've actually started stratifying them into 3 bins, small, medium, and large, according to the area of the tumor. And if you combine them all together, which the data I showed you is, we still get a clear defect. But what we started to do is look between P55 and P75 for just tumor growth, or P55 to P95 in our medium-sized tumors, because what we've seen is the larger the tumor, the quicker the animals seem to die, which is maybe intuitive. And when we basically create a ratio of the tumor size at 95 to 55 as a proxy for growth, they do seem to stay at like a ratio of 1, indicating a lack of growth, or even shrink a little bit below 1. So while we haven't gone through and actually like fact sorted them, or for GFP, and then looked at like H3P or some other indicator of proliferation, we do see that the tumors themselves, at least in the medium size initial tumor group at P55, don't seem to grow as much as the untreated vehicle control treated guys do. I hope that answers the question.
We have further questions as well. So a very nice talk and findings. Does targeting these pathways have applications in limiting pregnancy and regrowth after surgical resection in glioblastoma?
I didn't hear the first one. Of limiting what and regrowth? Limiting
dormancy and regrowth.
Yeah, that is also an excellent question, and frankly, we're not sure. Like, we have people are using BET inhibitors for various different tumor types right now, and the problem with glioblastoma and surgery is it's so, and I hope you could appreciate that even from the IUE model, is it's so diffuse and invasive. It's incredibly difficult to get all the tumor removed with surgery. But the common frontline treatment is surgery, radiation, and chemotherapy all at once, but we're only hitting these mice with chemotherapy. So I'm not sure if it would do anything to affect like time after the fact, but I think it's always going to come back. It's just, are we buying the patients more time or not? And at this point, say if this actually holds true, it might, be able to provide a little bit more time. But I don't think anything that we've seen is going to be able to actually stop it.
Cool. All right, so this is a little bit of a long question. So it's 2 questions from Sophie. Can you tell if angiogenesis is really happening in your model? Another explanation could be that tumor cells co-opt pre-existing vessels, so they grow around the pre-existing vessels without any angiogenesis happening. That's question number 1. And
yeah, so I'll address that one first. Yeah, so vascular mimicry may happen. What we showed in the first paper is, at least by CD31 and GFP double stain, that we are actually, they are endothelial. That's not to say that they're not contributing to parasites or some other third cell type that's enwrapping around the vessels, but by FACS and gene expression profiling and by immunohistochemistry, they all have an endothelial true signature. Again, in the RNA-seq, where we actually sorted for the cells, they have a proangiogenic signature. So whether it's actually true sprouting, no, we haven't actually gone through and seen that, but at least by FACS, immunohistochemistry, by the 3D imaging that we've seen with lectin perfusion, which will be on the inside of the vessel, right? And by slicing through the vessels and doing immuno for CD31 and GFP, and for the RNA-seq, and for single cell RNA-seq, we do think that they are true endothelium, but it is a really, really low concentration or amount. Like, the overwhelming majority of the vessels normally, at least in our patients and in this, are TAVs and not TDEX. I mean, we're talking less than like 1% probably across all experiments do you consistently see a TDEC contribution.
Cool. The second question was also about TDEX. So is it possible that the GFP positive endothelial cells resulted from endothelial cells being infected with the virus?
Yes, certainly. They could have, but then we would anticipate that they also got the, from a theoretical standpoint, yes, the glass promoter could have been electroporated and integrated the cargo into an endothelial cell, but then assuming that all the DNA together is electroporated, that endothelial cell would have lost P53, NF1, and PTEN, so you'd expect some kind of hemangioma or other kind of tumor. At least I would, but that's not to say that it's impossible, certainly.
Great. So there are think we're running out of time for questions. There are some other questions in the question box, Josh. So if I think Bernadette will email those to you, and if you'd like to answer those offline, that'll be great. So I think right now we're going to transition over. That was an exciting talk. Thank you, Josh. We're going to transition over to Zhanna. So I'd like to introduce Dr. Zhanna Nepiyushchikh, who will present her talk entitled Mitochondrial Bioenergetics of Collecting Lymphatic Vessels in Health and After Injury. Zhanna is a research scientist at the Petit Institute for Bioengineering and Bioscience, School of Mechanical Engineering at Georgia Institute of Technology. She works in the Laboratory of Lymphatic Biology and Bioengineering. Zhanna received her Doctor of Medicine degree from the State Medical Academy in St. Petersburg, Russia. She pursued postdoctoral training in the Department of Medical Physiology at Texas A&M School of Medicine, where she focused on the role of contractile and regulatory proteins involved in the phasic and tonic lymphatic contractile activity. She joined the Laboratory of Lymphatic Biology and Bioengineering in January 2014, and her research focuses on molecular aspects that control collecting lymphatic vessel contractility and mitochondrial bioenergetics in response to the dynamic changing mechanical environment, reactive oxygen and nitrogen species in health and pathology. So thank you, Zhanna, for joining us. I'm going to let you take it away.
Good afternoon, everyone. And I want to thank NAVBO committee first for this opportunity to present our data. And I will start right now. So title of my presentation, Mitochondrial Bioenergetics of Collecting Lymphatic Vessel in Healthy and After Injury. So a little with the introduction about lymphatic network. Lymphatic network has collecting lymphatic vessel and initial lymphatics. And my talk today will be about collecting lymphatic vessel. Collecting lymphatic vessel complex structure. It has adventitia, mostly it's collagen. It also has dendritic and macrophages in the wall. And main structure of collecting lymphatic vessel is lymphatic muscle cells and endothelial cells. So as I told previously, it's complex structure, and lymphatic muscle cells collecting lymphatic vessel is both cardiac and blood vasculature proteins, and lymphatic vessel exhibit robust phasic and tonic contraction. So if lymphatic vessel, collecting lymphatic vessels fails, it leads to disease called lymphedema. In lymphedema, this accumulation of proteins, fluid, and fat in the tissue, and it usually it can be genetic, or in developed country like US, it's most complication after breast or prostate cancer surgery, and it also can be because of parasite inside of lymphatic vessels. So the main goal of this study to evaluate metabolic state isolated collecting lymphatic vessel in healthy and pathological states for future diagnostic and therapies for preventing and treating lymphatic pump failure. We used a rat model for this study, rat popliteal lymphatic vessel that run on lateral part of rat hind limb. So popliteal lymphatic vessel was isolated, cleaned from surrounded tissue, perfused to remove rest of lymph, and we used HLN seahorse analyzer to calculate bioenergetic parameters. And mainly in this talk, in this presentation, I will talk. We used 3 tests: meter stress test with electrochain inhibitors, glycolysis stress test, and full dependency test. We also denuded lymphatic vessel to separate input of lymphatic muscle cells and lymphatic endothelial cells. So first one, I will talk about meter stress test. So we used different inhibitors for different electronic chain and calculated some parameters at the end of experiments. So I want to focus your attention here. This is naive lymphatic vessel, it's not injured. So after we removed endothelial cells, we see denuded lymphatic vessel demonstrate almost 50% decreased basal and maximal respiration, and it almost eliminate proton leaks. ITP production in intact and denuded vessels was not changed. We also defined that red popliteal lymphatic mitochondria is able to utilize almost all available fuel for respiration in a different degree, but there is no significant difference in between each fuels. We used red lymphatic surgical lymphatic injury model to injure lymphatics. We removed lymph node and connected to lymph node lymphatic vessel, and I point part of lymphatic vessel was left intact. And after 3 weeks, intact lymphatic vessel was isolated and used for the study. To monitor how contractility of lymphatic vessel changes during of time, we utilized near-infrared imaging system, and we saw by week 3 frequency of lymphatic vessel contraction significantly increase in injured leg, and also pocket amplitude, and not also pocket amplitude was significantly decreased. So we also used immunohistochemistry to see what the changes in area of in the surgical lymphatic isolated from surgical area, and we saw upregulation of inflammatory marker like TGB, upregulation of eNOS, and we also see downregulation of defense, oxidative defense, SOD, and we also seen oxidative stress damage in these lymphatics. We used sham and injured lymphatic vessel for evaluation of mitochondrial respiration and ITP production. And what we saw, injured red popliteal lymphatic vessel shows significant increase in maximal respiration, and it tends to increase in ITP production. We also denuded sham in injured lymphatic vessel and to evaluate mitochondrial respiration and ITP production. And what we saw, maximal respiration was decreased in both vessels. Injured popliteal lymphatic vessels showed significant increase in vessel respiration and significantly decreased in proton leaks. Glycolysis stress test not shows any significant changes in glycolysis in both sham and injured lymphatic vessels. And for conclusion from our study, so I want to say we work in progress right now, and have more information. But to summarize this part of our study, I would say lymphatic muscle and lymphatic endothelial cells almost equally contribute to mitochondrial respiration and ITP production. Lymphatic vessel mitochondria is able to utilize all available fuel to support respiration. Lymphatic endothelial cells significantly contribute to proton leak in whole lymphatic vessels. And lymphatic endothelial cells possible involved in increase of respiration and oxygen consumption in injured lymphatic vessel, probably to produce nitric oxide. And mitochondria of lymphatic vessel are capable to detect early cellular insult and orchestrate inflammatory response. And decreases of oxidative defense, sodium oxidase dismutase in mitochondria, possibly switch generation of nitric oxide to toxic peroxynitrite, and it might be a cause of damage in lymphatic vessel, as we see in our model. And in the end, I want to say thank you, my graduate student and undergraduate student, especially Rachel Chin, who is undergraduate student. She's working actually on her paper, and first author paper, and she significantly contributed in the beginning of this study. Sophia Maurice, she's PhD student, and she's working as first author of injured model paper. And I want to say thank you all my lab members and also audience for your attention, and I'm ready for any questions.
Awesome. That was a great talk, Zhanna. We have a few questions already in the question box, so if anyone has any other questions, feel free to add them now. The first two questions are pretty similar. So the first question is very interesting talk. How did you denude the lymphatic vessels? And the second set of questions, I think, are about the same thing. It says, can you clarify again the nature of the lymphatic injury used? Was this air embolization to remove endothelial cells or some other injury? Presumably some injury occurs just with the surgical isolation of these tiny lymphatics. So.
First question was how we denuded lymphatic vessel. So we denuded it with air ball. We passed air ball, connected isolated lymphatic vessel to through glass cannula to small syringe filled with air, and during 10 seconds we passed this air through lymphatic vessels, and this technique was published in several papers, was used to denude lymphatic vessel. And second question, it's possible, I don't know, second and third question, it's possible to injure lymphatic vessel during air passing, but I don't know any technique that can safely remove lymphatic endothelial cells and don't cause of damage of lymphatic vessel by itself. But we're actually working on another technique to isolate endothelial tube. It might be more precise to study input of lymphatic endothelial cells in bioenergetics. And maybe I missed something.
I can say. So I think the second question was asking about the what was the nature of the lymphatic injury that was used. But I think you were removing the lymph node. Is that right?
Lymph nodes. I will come back to my slide. So we removed lymph node, popliteal lymph node, and partially lymphatic vessel connected to this lymph node.
Cool. Okay. So I think that addresses the second question. And then we have another question about the SOD, the decrease in SOD2. They said the SOD2 decrease is very interesting. Can you try to treat mice with mitotempo, SOD2 mimetic, to test the role of SOD2 impairment?
Yeah, we have, yeah, thank you, very good question. We used mitotempo to, in some study, to see how it affects in a general contractility. And we saw interesting, so short answer, we didn't do it not yet in this model. I think it's a future, our future direction. But to answer about mitotempo, we saw mitotempo actually by itself affect lymphatic vessel contractility. So it's not surprise, but it's interesting. So I think like reactive oxygen species not so bad for contractility, and they're involved in normal contractile pumping activity of lymphatic vessel. Yes, but thank you for this question. Yeah, it's our future direction to find a way that can restore mitochondrial function in damaged lymphatics.
Cool. And I think that's all the question in the chat box. I had one more question. So it looked like you were assessing the lymphatic contractile function in vivo, right, with near-infrared tracers. So have you tried also maybe surgically isolating the lymphatic vessels, like how you do for the mitochondrial respiration experiment, but then cannulating it onto two glass pipettes and then studying the contractile function?
Yes. Yeah, we're working on this. I couldn't include it in the presentation. We have a small number for this study. Yes, we do.
Right. Nice. Pretty fascinating, and it seems very relevant to patients with lymphedema. So it's an awesome talk. I think that's all the questions for now. So thank you, Zhanna and Josh, for being with us, as well as everybody else for tuning in. If anyone else has a question, please email info@navbo.org, and we'll make sure to share your questions with our speakers, and they'll get back to you via email. So I hope everybody enjoyed today's webinar. Please look for more information about future events on the NAVBO website. And of course, we hope you can join us at Vascular Biology 2023 in Newport, Rhode Island. This is the slide on that. So please visit the NAVBO website to register or use the QR code on your screen. And thank you guys for joining us.